continuing on with chapter 1, after an experiment, you might have, after talking about those experiments, you might have the question, okay, so we've got hypotheses, but when do hypotheses become theories? Or how are hypotheses and theories different from each other? And yeah, by the way, what are theories? So let's talk a little bit about what those words are and what they mean. So we talked about a hypothesis, right? We use those in experiments. A hypothesis is a proposed explanation for a phenomenon. So we use these, for setting up things like an experiment like we talked about. This one is going to be used for, leading to testable predictions. So an example of a hypothesis is, I've got a hypothesis about why there's less traffic on Friday mornings than Thursday mornings. And then you can lead into testable predictions about that, right? On the other hand, you've got theories. So hypotheses, they're very narrow in, scope. So you could also say, something about like squirrels. Squirrels, you've made probably plenty of observations about squirrels. They climb trees, they dart around, they eat nuts, and so forth. And so then you could make a hypothesis about, that squirrels bury nuts because they are hiding them from other squirrels. And so then you could make that into a testable prediction that you could then create an experiment about. Theory, on the other hand, a theory is not a speculation or a guess. So a lot of times people use the word theory that are non-scientists as a speculation or a guess. I have a theory about why John never has any money in his pocket to pay for dinner, so it's not a speculation or a guess. They are hypotheses of which scientists are most certain. Basically, a theory is a group of hypotheses that have been put together that are repeatedly tested and supported. So these are basically the things that we are most certain of in science. They are broader in scope than hypotheses. So hypotheses are very narrow, talking about single species or, a small section of, an ecosystem or something like that. On the other hand, theories are much broader in scope because you can think about it, I always think about it like a theory is like an umbrella that a bunch of little hypotheses are hiding under almost. Because a theory has been put together from a bunch of different hypotheses. So what are some theories that you're familiar with? I'm sure you're familiar with some theories. Probably the one that comes to mind is the one we've already talked about, which is the theory of evolution. And the theory of evolution has tons of hypotheses in it, right? So an example of a hypothesis that is tied to the theory of evolution is every time we put together a fossil history of a group of organisms, because that's our best testable prediction on how those organisms are related to each other. But it is related to the theory of evolution because it is a chunk of evidence that is working to support that theory of evolution on how organisms change over time. So other theories that are well known in science that we also will be talking about later on, we'll be talking about the cell theory. We'll be talking about the endosymbiosis theory. You may have also mentioned things like the theory of gravity. There's the theory of gravity and the law of gravity. They're both actually different. One is mathematical and the other is descriptive. And you may also have talked about the Big Bang Theory, string theory. All of these are big, broad ideas that are supported by tested hypotheses. Okay. So these were supposed to pop up one at a time. Not sure what happened. This is related to the experiments that we talked about in the previous lecture. I'm going to talk a little bit about the different groups that showed up in those experiments. So first, the treatment, that is any type of experimental condition that you applied to individuals. So for example, in the echinacea experiment, giving echinacea to the groups, that is the treatment, right? Because it's what you're changing for the experiment. If, for example, you are growing plants and you decide to give salt to some of the plants, but not the others, salt would be the treatment. On the other hand, one of the things that is really important as far as designing an experiment is that you need 2 different sets of groups at a minimum. Now, there were 4 groups in the other experiment on that echinacea, but it really came down to that were basically 2 groups. One is the experimental group. The experimental group is the one that you do the treatment to. So in that echinacea experiment, the experimental group, or in that case groups, are the ones that were given echinacea. Whether they were given echinacea before or after virus exposure, they were the ones that were given the echinacea because echinacea was the treatment. Control groups, these are important because these are a group of individuals who are treated identically to the experimental group, except they're not given the treatment. So they are, in the echinacea group, they're the ones that got the sugar pill. So they are given the exact same sets of conditions, but they're not given the actual treatment. And so that's why we actually give them, in that case of the echinacea treatment, a sugar pill. So that way they're still getting a pill, but it just doesn't have, in that case, the echinacea in it. We do the same thing with any sort of medical trial. So for example, the vaccine trials that have been going on for COVID, the control groups would have gotten an injection, it just would have been saline, no actual components of the vaccine. So that way they were given everything except for the active component of the vaccine to create the immune response. So control groups are incredibly important because they allow us to find out if there is actually an effect or not. So let's take a look at that.